Hello students, the topic of today's video lecture is DNA replication process. In DNA replication process, four steps are followed. Number 1, replication fork formation. Second step is primer binding. Third step is elongation and the fourth step is termination. In today's video, we will discuss these four steps in detail. So the first step in DNA replication process is replication fork formation. In this step, before DNA can be replicated, the double-strand molecule must be unzipped into two single strands. Basically, DNA has four bases called adenine, thymine, cytosine, guanine that form pairs between the two strands. In order to unwind DNA, these interactions between base pairs must be broken. This is formed by an enzyme known as DNA helicase. DNA helicase separates the strand into Y shape, known as replication fork. Now, the second step is primer binding. In this step, the leading strand is the simplest to replicate it. Once the DNA strand has been separated, a short piece of DNA called a primer binds to the three end of the strand. The primer always binds as the starting point for the replication. Primers are generated by the enzyme DNA primase, and the DNA polymerase 3 can only nucleotides to existing strands of DNA. In elongation step, enzymes known as DNA polymerases are responsible creating the new strands by a process called elongation. Uh, in eukaryotic cells, polymerases alpha, delta, and epsilon are the primary polymerases involved in DNA replication. Because replication proceeds in the 5 to 3 direction on the leading strand, the newly formed strand is continuous. The lagging strand begins replication by binding with multiple primers. Each primer is only several bases apart. DNA polymerase then adds these pieces of DNA called Okazaki fragments to the strand between primers, and this process of replication is discontinuous as the newly created fragments are disjoint and involves the addition of new nucleotides based on complementary of the templated strands. The daughter strands is elongated with the binding of more DNA nucleotides. Okay, in the last step, which is called termination, once both the continuous and discontinuous strands are formed, an enzyme called exonuclease removes all the RNA primers from the original strands, and these primers are then replaced with appropriate bases. Another enzyme called DNA ligase joins Okazaki fragments together, forming a single unified strand. The ends of the linear DNA presents a problem as DNA polymerases can only add nucleotides in the 5 to 3 direction. Once completed, the parent strand and its complementary DNA strands coils into the familiar double helix shape. In the end, replication produces two DNA molecules, each with one strand from the parent molecule, and one new strand is formed.